Hi, everyone. I'm very pleased to be the co-chair of this wonderful symposium on innovative brain recording methods in cognitive and social neuroscience. I'm Sarah Lippe. I'm full professor at the psychology department and scientist at Saint-Justine in Montreal, and I direct the Neuroscience of Early Development Lab. I co-chair this symposium with Dr. Anne Gallagher, associate professor at the psychology department and scientist at Saint-Justine. She directs the neurodevelopmental optical imaging lab. We are very pleased to have four amazing speakers for this symposium. The first speaker is Ole Hansen. He's full professor at the School of Psychology of University of Birmingham. He is the co-director of the Center for Human Brain Health. He is a world-leading expert in the research aiming at uncovering the mechanisms of neural oscillations for cognition, and he uses especially magnetoencephalography. Our second speaker, Dr. Guillaume Dumas, is assistant professor at the Psychiatry and Addictology Department at the University of Montreal and associate academic member of the MIRA. And his research innovatively combines computational biology and social neuroscience approaches. Dr. Susan Dicker is a senior research scientist at New York University, Max Planck Center for Language, Music, and Emotion. And she developed extremely well-thought citizen neuroscience project to study brain and behavior into real-world environments. And Dr. Joy Hirsch, professor of psychiatry and of comparative medicine at Yale School of Medicine. Currently, her research uses EEG and NIRS to study neural operations that regulate interpersonal dialogue and social interactions. So as you know, joint attention has been one of the underlying mechanisms driving social interaction. And thanks to our amazing speakers, our symposium will provide very informative content on the dynamics of the brain in several settings of incremental complexity. So first, Dr. Hansen's lecture will focus on the brain dynamics of attention, innovatively using the invisible rapid frequency tagging in MEG. And then Dr. Dumas, Dicker, and Hirsch will explain the recent developments in the investigation of brain dynamics in social interactions, from joint attention to interactive dyads and group dynamics in classroom and even museums. So for Dr. Gallagher and I, their studies are particularly interesting and valuable in order to better understand atypical social adjustments in neurodevelopmental disorders. So thank you again to our amazing speakers. And I think there will be time at the end of the symposium to answer a few questions.
Hello, my name is Ole Jensen. I'm going to present the talk using invisible rapid frequency tagging to investigate attention and speech gesture integration. So this is work primarily done at the Center for Human Brain Health. This is a new brain imaging center at University of Birmingham. We opened a few years ago. So what I'm going to tell you about first is something about brain oscillations, more specifically the alpha rhythm, and how we investigate that using rapid frequency tagging. So over the years it has been discussed what this alpha rhythm is doing. It was first reported in the late 1920s by Hans Berger, and it's sort of the strongest spontaneous rhythm you see from the human brain. Initially it was thought that this rhythm reflects a state of rest or idling, but later we and others have been able to demonstrate that this rhythm indeed reflects inhibition of specific regions. So what we have been doing is to over the years is to develop this idea. We have a paper here we had out in 2010 which put forward that this alpha activity reflects inhibition and can serve to gate information flow in the brain by inhibiting regions not involved in the task. And by this, there's also an implication that the alpha oscillations could be doing gain control. So in order to investigate that further, we have been developing this technique called rapid invisible frequency tagging. So basically the idea is that using this new type of projector called a ProPEX, we can flicker up to 1440 hertz in a visual scene. So we can literally control every pixel in a visual display with very high temporal resolution. And then we can measure the brain response, in this case with MEG, and we use MEG since it allows us to locate where in the brain the neural activity is generated. So frequency tagging is nothing new, but however typically is used around 10 to 20 hertz. What we have been able to do is to use frequency tagging around between 50 to 80 hertz. The main advantage is that we still get a sizable response, but the visual input cannot be seen. The flicker cannot be seen, and we can measure changes in excitability with a very high time resolution. So we have been using this to investigate gain control in the brain. By gain control, I here mean the excitability in early visual cortex and how that is modulated by spatial attention. So this is a very complicated diagram, but it's to get across that we have a very specific idea about how the alpha oscillations exercises a top-down drive that then sets the excitability of early visual cortex. We have explored that hypothesis using rapid frequency tagging. As it turns out, this hypothesis does not seem to hold up. So how do we use rapid frequency tagging? Well, here's an example where we ask people to attend to the left or the right, and then we are flickering the left item, in this case the broccoli, at 63 hertz, and the right item, the ice cream, at 78 hertz, and then we can measure the response from the brain using MEG. Right, and we get quite a sizable response since the noise floor is quite low at these high frequencies. And what we now can do is to go in and play with, say, spatial attention, and then ask how does this frequency tagging signal change with the allocation of attention. So the first paper I'm going to talk about is some results found by Alexander Segalov. Is already out in human brain mapping, and there we have been looking at these alpha oscillations to specifically address how they reflect gain control in the early visual cortex. And in this case, we didn't flicker the signal at a narrow band, but we flickered the signal at a broad band, in this case from 60 to 70 hertz. We had two stimuli, left and right, and a fixation cross. People had to attend to the left or the right stimuli. What you see down here is an example of the input signal. So we have two flickering signals that for the left and right stimuli, they are orthogonal, albeit the power spectra looks similar, right? So what we then also have been doing is to calculate the cross correlation between the input, the broadband input, and the MEG response. And what you see here to the right is a measure of this cross correlation, and after the stimuli turns on, you get an increase in this cross correlation. And furthermore, when people attend to an object, you get a stronger cross correlation compared to unattended. So this cross correlation is a reflection of excitability in early visual cortex. We can also measure the delay by looking at when this cross correlation is peaking. Okay, so here are the first results using that technique. So in the upper panels here, we have examples from a participant or participants, a group of participants getting stimulation in their left hemifield, and we then measure the response by the cross correlation. And since we stimulate over the left in the left hemifield, we get the response to the right. And this is also what you see here on MRI, where we have superimposed the source modeling. Contralateral to the stimulation, we get the response. Furthermore, what you should also appreciate is that there's an increase in this response with attention, and that's something we have quantified further. And of course, when we stimulate in the right hemifield, we get the opposite response. So what you see here is then the frequency tagging signal with respect to attention, and what you should appreciate is that there's a stronger response for the attended than the unattended objects, as one would expect. So the frequency tagging indeed indexes the excitability associated with attention. And we can also look at these attention effects, and they appear in early visual cortex. So then you might ask, how does that relate to the alpha band activity? So the beauty of doing the high frequency tagging is also that we don't disturb the lower frequency oscillations. So what you see here is that the alpha oscillations, they are attenuated to the right when we ask people to attend to the left, and vice versa. So this is an old finding reproduced in numerous studies. We can also source localize this modulation in the alpha band activity, and the sources appear in parietal occipital cortex. So what we now can do is to compare the frequency tagging response and the alpha response, or the modulations of these signals. And what you already can see here by the source modeling is that the modulation of the alpha oscillations is slightly downstream compared to the modulation of the frequency tagging signal, right? And we can further quantify that by a statistical test, and indeed there's a significant difference between where the tagging signal and the alpha modulation shows up, right? So this here suggests that the sources of the alpha activity and the sources of the frequency tagging are at different locations. So this is of course important because it sort of does not fully resonate with the notion that the alpha activity is exercising control over the neuronal excitability as reflected by the frequency tagging. So what we now have done next is directly compare the modulation of the alpha power to the frequency tagging signal. So we simply did this by making use of the natural variation in the alpha signal over trials. And what you see here on the top is the alpha activity recorded ipsilateral to the intended direction. And what you should appreciate here is that that we can split the alpha power, the trials, according to high and low alpha power. And then for these respective trials, we can look at the frequency tagging signal. And lo and behold, what we find is that the frequency tagging is exactly the same for the trials with low alpha power as compared to trials with high alpha power, right? So this here actually suggests that the alpha power is not modulating neuronal excitability, right? So this has quite important implications. Namely, it suggests that the alpha power and neuronal excitability are uncoupled. So it also implies that we might have to give up on the idea that the alpha oscillations are implementing gain control. So what is going on? It is not that alpha power is unimportant. Rather, we think that gain control is implemented by other mechanisms in early visual cortex, and then there's a feed-forward flow of visual information, and that feed-forward flow is then being gated later on by the alpha band activity, right? So it's like the alpha activity is sort of opening and closing the door to the information flow, but it does not do that by modulating the gain control in early visual cortex. So to make that crystal clear, imagine here there's this guy, it might be my son, playing his electrical guitar. I find it a bit too loud, so what can I do? Well, I can go to his room and turn down his amplifier. That's one possibility. That would be gain control. But what I can also do is simply to close the door to his room. That would be gating, right? So as it turns out now that we think that the alpha activity is doing the gating, namely corresponding to closing the door rather than the gain control. Okay, so I will move on to the next study. This is again using rapid frequency tagging in a cognitive task, in this case to investigate auditory and visual information integration. This is a study spearheaded by Linda Drivers, and it was just out in human brain mapping. So the idea about this study is that we have been presenting these snippets of a person gesturing. So at the same time, we also present words to that to the participants. So the participant can listen to these words while watching the person here gesturing. So we also have a condition where it's difficult to hear the spoken words. So we basically degrade the speech to sort of simulate a noisy situation. So the question now is, can the gestures support the comprehension of the words, right? So it could be that the participant hear the word drive, and then will the participant then see the person sort of gesturing drive, that should support the comprehension, in particular in a noisy setting. And this is indeed what is going on, because that's how we designed the study. What I should also mention is that we did frequency tagging of the visual input here at 68 Hz, but also the auditory input, the speech, we modulated that by 61 Hz, right? So then we can investigate both the frequency tagging response to the auditory and the visual modality and the integration. So this is the behavioral data. What you should appreciate here is that when we have degraded speech and we have a gesture that does not match with the speech, while then accuracy is fairly low. However, if the gesture matches the spoken words, then the comprehension is higher, right? So this here shows that our study works in a sense that the gesture can support can support the comprehension of the speech, in particular when the speech is degraded. Okay, so what happens at the brain level in terms of integration? To investigate that, we looked at these frequency tagging signals. So I should mention that the frequency signals were, the frequency tagging was phase locked to the onset of the stimuli, both for the auditory and the visual input. And we can then average these trials together, and even with the naked eye, we can see these frequency tagging responses. This is just to demonstrate that it works. So on the top plot is the example where we look at sensors located over auditory cortex, and what we see, lo and behold, is a strong 61 Hz response, which is stronger when we stimulate as compared to the baseline. When we look over visual cortex, we see a 68 Hz response that is also stronger than the baseline. So we get an auditory signal and we get a visual signal as reflected in these frequency tagging responses. Okay, now comes the issue of integration. So now we are making use of a trick that also has been used in other frequency tagging studies, namely by looking at the intermodulation. So in this case, we have the auditory input at 61 Hz and we have the visual input at 68 Hz. So now it turns out that if these two rhythms are interacting, there will be a modulation at the difference and at the sum of these frequencies. So in this particular case, we should be able to see a peak at 7 Hz. This would only occur if there's a nonlinear interaction between these two signals. So just a simple super, if these two signals are superimposed and add up at the scalp level, we will not see a 7 Hz peak in the power spectrum. However, if there's a nonlinear integration reflecting sort of neuronal interactions between auditory and visual signals, we will see a peak at 7 Hz. Okay, so what's happening? Well, lo and behold, when we look at the data, compare the power spectra during the stimulation for baseline, we do see an increase at 7 Hz. Furthermore, we see this increase strongest over sort of left brain regions, so which could reflect the engagement of the language system, right? So our first manipulation is working. We do get a fingerprint of the audiovisual integration as reflected by an increase in this 7 Hz peak. Okay, so what is going on in terms of the different task conditions? You might have expected that it is in the degraded condition where you have the match, where you do the sort of strongest audiovisual integration, right? Because that's where you really benefit from the visual input. Nevertheless, this is not what the data shows. The data shows that you get the strongest 7 Hz peak when there is a clear speed signal, together with a visual input, and these two they match. So that's when you can have the sort of strongest congruency between the auditory and visual input. That is when you get the strongest integration effect in the left language region. So this is the core finding of this study. So I'm going to summarize here and to conclude. We have now developed a new paradigm using rapid frequency tagging. So it allows us to investigate, first of all, the excitability in the early visual cortex. The main advantage of this technique is that it's invisible. People do not see the flicker. Furthermore, it does not, the frequency is so high it does not mess with oscillations at slower frequencies in the alpha and beta range. So that allows us to investigate the interaction between neuronal excitability as indexed by the frequency tagging and the ongoing oscillations. Furthermore, we have been able to show that this rapid frequency tagging can be used to investigate nonlinear integration, audio-visual integration. So the core findings when applying this to cognitive paradigms is that, to our big surprise, we do not see that the alpha modulations are modulating neuronal excitability in early visual cortex. This has the important implication that the alpha activity might not be doing gain control in early visual cortex as we initially hypothesized. Rather, the alpha activity might be involved in doing gating slightly downstream. Furthermore, we have been using the technique to do investigate audio-visual integration, in particular speech-gesture integration, and what we found was an effect of integration in left prefrontal cortex. So we also want to expand on these studies to further study social interactions in the context of audio-visual input. I'm going to end here and acknowledge the core people involved in doing this work. The first study was done by Alexander Sigalov. He did the work on alpha oscillations and rapid frequency tagging. The second study was done by Linda Drivers, and she did the work on audio-visual integration, also using frequency tagging in the auditory and visual domain. And finally, I should acknowledge the funders being involved in supporting these studies. Thank you very much for your attention.
Hi, I'm Joy Hirsch, and a professor of neuroscience at Yale School of Medicine. And I'm delighted to be here to participate in this international organization of psychophysiology. I'd like to thank the organizers of the symposium for inviting me to give this talk. Our symposium is an extremely exciting topic, I think, of innovative brain recording methods for cognitive and social neuroscience. And to that end, I'm going to talk about the advances that can be made from hyperscanning with near-infrared spectroscopy. And that brings me really to my topic of the emerging theoretical framework of two-person neuroscience. And this is based on work that is done with interactive functional near-infrared spectroscopy. And we see a picture of such a setup here, where two people are sitting across from each other wearing caps with detectors. And these detectors are the basic elements of the optical imaging techniques that measure both absorption of oxyhemoglobin and absorption of deoxyhemoglobin by wavelets of light that are injected into the brain. These measurements serve as proxies of neural activity, similar to the hemodynamic responses that are observed conventionally with functional functional MRI. We're just doing it with optical methods rather than by interference methods that are used for functional magnetic resonance imaging. This technique, as you can see, without the bore of the scanner and with the high magnetic field, enables simultaneous imaging during live interactions, and it enables many additional techniques to be acquired simultaneously because of the absence of the high magnetic field. So we can add eye tracking, facial recognition cameras. As you can see, some of our cameras up here hanging from the ceiling. We can do behavioral measurements with dials that we have on the table separating the two participants. We can interleave EEG electrodes into the caps so we can acquire EEG simultaneously, of course, microphones and speakers. Now, why would we want to do this? Well, one of the first reasons is that humans are profoundly social and interactive in nature. And most of our neuroscience investigations up to this point have involved single brains. And so we've learned a great deal about imaging, about the brains of individuals, but we know very little about the brains of dyads, that is, two individuals interactive in a dynamic encounter. And this, of course, is due because of the limitations of a technology that's been focused on single individuals. But with this new technology, we can break through a lot of those barriers and look at the brain in interactive modes. Now, of course, there are limitations to this. Our limitations include relatively coarse resolution. We're imaging at approximately 3 centimeters. Now, some techniques using near-infrared spectroscopy can improve that resolution almost an order of magnitude. But nonetheless, we still are imaging only responses from a superficial aspect of the cortex. We can't image the internal functions of structures in the brain. So we're limited to approximately 3 centimeters of depth into the cortex. But nonetheless, the main advantage here is that we're able to image two persons interacting and perhaps address some of the really important questions of the brain in interactive modes. So let's think about what those questions might be. From philosophy, we have the interactive brain hypothesis that suggests that two dynamically interactive brains engage dedicated neural systems that are distinct from single non-interactive brains performing the same task. Now, this idea has been around for a long time, but it hasn't really been tested rigorously until the opportunities provided by functional near-infrared spectroscopy have come online. So what I'm going to tell you about today is a couple of opportunities to test the interactive brain hypothesis. And the first one is with language. So the interactive brain hypothesis for people speaking to each other is that we would expect that talking in a two-person dialogue would activate different neural systems than talking and listening in solo mode. Particularly, we would predict that these differences would be in either Wernicke's area or Broca's area, or both, because those are the canonical language regions, and we can call upon the wealth of information we have about the receptive and productive functions, respectively, of these areas. So here we design an experimental paradigm using near-infrared spectroscopy to test these hypotheses using a standard task of object naming. Essentially, this has been used conventionally in functional MRI studies of receptive and productive language. But here we do it for two people. So we have a situation where a participant is shown a picture of an object, and that person has 15 seconds to talk about that object, and the participant simply listens, and then the next person gets an object, in this case a camera, 15 seconds to talk, and the other participant just listens. This, what I just described, is the solo mode, talking and listening, but no interaction. In the interactive case, the object is presented, the individual talks about it, subject listens, but then the next subject responds and says, makes a comment about what the first person has discussed with respect to the object, in this case, a clock, and then presents her picture, which is a camera, discusses it while the participant listens, but then before the participant responds, talks back to the person about the camera. So we have the same task, talking and listening, in one case in solo mode, and the other case in an interactive mode. And so we can describe the interactive and the solo modes here, where people are just talking and listening in interaction and without interaction. And in this case, the results are very, very clear. In the interactive dialogue case, Wernicke's area is much more active than in the non-interactive monologue case. And so these findings are consistent, at least, with the hypothesis that interaction matters with respect to language processing, at least in Wernicke's area. But now we want to test this same, in this case, yes, I forgot to mention, Broca's area doesn't care whether you're talking to yourself or whether you're talking to somebody else. It appears in this paradigm, under these conditions, to be particularly interested in production and not really interaction. The interaction seems to be a property of the back of the brain, the temporal parietal junction on the left side, commonly known as Wernicke's area. More details of this study have been published, as indicated here, in SCAN, Social, Cognitive, and Affective Neuroscience. If you would like to look it up for more details. So we can go on and test other aspects of this hypothesis using face-to-face contact. And again, nothing verbal, just face-to-face interaction. And we would predict that the real face-to-face viewing processes are different from, say, simulated face viewing processes, meaning either pictures or videos or even a robot. So here we have a case where people in the interactive mode simply gaze at a partner's eyes, or in this case, gaze at a video face of eyes of an individual. And the data are, again, consistent with the interactive brain hypothesis when we contrast the real face greater than the video face, and we see a large cluster of activity in the right temporal parietal junction, and particularly involving the supramarginal gyrus or the superior temporal gyrus, regions known to be consistent with the social brain. Again, these findings are published in Frontiers of Human Neuroscience, and also in neuroimage. We test this hypothesis again with the confederate, in this case, being a robot with a human-like face and eyes that have some human-like qualities. And again, the face-to-face greater than the robot gaze activates supramarginal gyrus and superior temporal gyrus, again, consistent with the hypothesis that realness in faces in interaction actually matter. Again, this paper is published in Frontiers in Robotics and AI, if people were interested. So just to summarize the interactive brain hypothesis using near-infrared spectroscopy, in both language and visual interactions between individuals differ from non-interactive. So there's quite a bit of support for the interactive brain hypothesis. And these differences tend to be observed in the left temporal parietal junction for language and the right temporal parietal junction for visual face processing. And finally, these emerging findings, these data and others that have been published since these studies, suggest that there's a new general domain of specialization that has not been appreciated prior to this type of work, that suggests that interpersonal interactions are specialized in temporal parietal junction regions. So interactions between individuals, the dyadic responses, are actually also a domain of the social brain. So we can test these hypotheses in other ways as well. And one of the advantages of doing live two-person interactive neuroscience is that we can test hypotheses about neural coupling. Now, we start with a neural coupling hypothesis that actually was first proposed by Yuri Hassan and colleagues. One of the first proposals of this hypothesis was in 2004, published in Science. And according to the hypothesis, that the cross-brain coupling represents the sharing of information between two brains. And so in accordance with this hypothesis and the paradigms that we have presented here, we would expect greater cross-brain coherence between regions that are specialized for a social interaction. And in our previous studies, we've shown that the back of the brain, temporal parietal junction, specialized for eye-to-eye contact and dialogue, again, back of the brain, the right temporal parietal junction would indeed be highly correlated with respect to social interaction. And indeed, that is what the findings show for both eye-to-eye contact and for interaction between individuals during dialogue. So the emerging theoretical framework for the interactive brain suggests, one, that live social interactions recruit neural operations that first code real in-person facial and verbal interactions. So there's specialization for the send and receive functions that are associated with social interactions. And two, that these brain regions that are engaged in simultaneous sending and receiving social information synchronize during the sharing of the reciprocal social information, and that these synchronies tend to include the sensory systems, visual and auditory, and back of the brain processes, as opposed to front of the brain processes, at least in the simple types of paradigms that I have outlined here. So all of this live social interaction information is available largely due to the advances in functional near-infrared spectroscopy as a neural imaging technique of choice, particularly when it's applied in a hyper-scanning environment. And so I think it makes an important contribution to the discussion of innovative brain recording methods for cognitive and social neuroscience. And so I hope that this work, along with others that are presented by my colleagues in this symposium, generate a great deal of interest, enthusiasm, and discussion. I'm sorry that we can't be there in person, because the discussion in person is a great deal of fun, but hopefully we can carry on online. So with that being said, I want to give credit where credit is due. Much of the work is done by research faculty in my lab, Adam Noah, Chen Zhang, and Mark Tide, who is a linguist who has helped us a great deal with our studies of live verbal interactions. Research assistants, graduate students, undergraduate students, visiting fellows, my colleagues at University College London, all contributed to the development and continuation of this work. So thank you for listening, and I look forward to hearing from you should you want to correspond by email or otherwise. Thank you very much.
Hi, everyone. It's a real pleasure to be virtually here, and I would like to thank my colleagues for organizing this symposium and looking forward for the interaction session at the end. So today I will speak about something that have occupied my research for the last 10 years, namely interbrain synchronization. And I will describe early observation of neurobehavioral correlations and try to describe also some paths toward causal explanation in the future. Those are like all the people I had the immense chance to work with, and I want to thank them and thank also the funding and support that allow this research to continue. And before getting to interbrain synchronization, let's talk about intra-brain synchronization, which has been a focus of lots of attention in neuroscience in the late 90s, early 2000s. And in this review by Francisco Varela called The Brain Web, they describe well how brain synchronization, neural synchrony, is a functional mechanism for large-scale integration of information. And so in this review, what was interesting is that they described the brain for the first time as a peer-to-peer network. So we were way before Bitcoin at that time, but they took the example of Napster, and showing how the brain is rather than a centralized processing unit of information like a computer, the network and in its distributed peer-to-peer fashion is more like this organic cell assemblies that coordinate the activity and make a merged function. And so this coordination of cell assembly at distance brain area or brain locations is illustrated here. So it's supported by anatomical backbone, but the dynamical retranscription of this coordination is this neural synchronization. And so when you go at the social level and that you have two brains that are interacting within each other through the environment, well, those two neural dynamics that are in the cell assemblies between the two brains, inside the two brains, are modulated by their coupling with the environment. And since there is this mutual coupling, they are modulating each other. And so they are all stimulus and receiver of this stimulus at the same time. And you have, through sensory motor coupling, interbrain synchronization that could emerge. But so that's more like theoretical, and concretely the problem was that initially the neuroscience of two individuals engaged in dynamical interaction was pretty scarce, and some people call it even the dark matter of neuroscience in the early days. And 10 years ago, it was indeed the case. And for the last 10 years, I've been working on how to operationalize this naturalization of social interaction by combining experimental work, computational modeling, even combining the two with a new paradigm called the human dynamic clamp, that is the human interacting in real time with the computational model. And we're going to start by talking about hyperscanning, EEG, and hyperscanning, because that's the main empirical approach for study of interbrain synchronization. So the term was coined by Reid Montague, and it comes from fMRI. So that's why we come from scanning to hyperscanning. And in my case, I started with behavioral hyperscanning, highly influenced by developmental psychology, and my supervisor at that time, Jackie Nadel, has brilliantly demonstrated that 3-month-old kids can decipher and disentangle videos of their parents live or pre-recorded, thus showing how we are already equipped at the neural level to disentangle those very fine-grained social dynamics, even at early age of 3 months. And what we did in collaboration with Jacques Martinery for the neurodynamical aspect is to synchronize two EEG systems while having this dual video, and ask people to imitate each other to understand what's going on when there is this perception action loop between two nervous systems. And we first show that at the intra-brain level, the role you are taking in the interaction and the context in which the interaction unfolds modulate your brain activity. And so basically, if you are imitator or model, or if you are in a spontaneous interaction or more like forced induced imitation, so for instance, like when we say here, participant on the left imitate participant on the right, and there is no way around, it's only that direction, is forced. While if we say you both move your hands and you imitate each other whenever you want, then it's more like spontaneous. And we show that in the case of spontaneous imitation, we have a co-ownership of action that is not present in the forced one. And it's characterized by a very symmetric neural pattern implicating the right parietal area. And you will see that's kind of connecting with other results in this talk. But the thing that is also interesting is that we replicate those observations with fMRI, showing that there is this double dissociation between social role and social context. And so if we take, for instance, the co-ownership of action during spontaneous imitation and this right parietal junction symmetric neural pattern at the intra-brain level. So actually we see here that there is increase of activity in the right parietal junction. But what we show also is at the interbrain level that there is a synchronization between those two brain regions. So basically that the neural dynamics of the two brains get synchronized in the right parietal junction during spontaneous imitation. And it's not only the symmetric part that actually we observe, we also observe an asymmetric part, thus showing that those neural dynamics and those interbrain synchronization also reflect the role that people are taking in the interaction. So here, for instance, model, the person that proposed a new movement, an imitator that received the movement. And we show that with language interaction as well, with Alejandro Perez, for instance, and with arrhythmic interactions such as affective touch. So in that case, with the team of Shamit Suri, with Pavel Goldstein, where we show that not only it's already interesting on its own, affective touch can diminish pain. We show that the diminution, the reduction of pain actually correlate with the level of empathy of the caretaker that hold the hands, and also that it's correlated with the interbrain synchronization. So that's interesting also because arrhythmic modulation kind of show that interbrain synchronization is not just entrainment phenomenon, because here you don't have rhythm, so there is no entrainment per se, but still you have this correlation with social bounding. And we show that also with attachment, so more like higher level social dimensions, that the relationship that you have with others, such as strangers, best friends, or romantic couple, also participate in the modulation of those interbrain synchronization. And it's also context-based, so that during a motor task or during an empathy-giving task, those relationships will affect differently the interbrain synchronization. So for instance, motor task is more linear. The more you are connected at the relationship and attachment level, the more you are synchronized, while for empathy giving, it's not linear. So, but that was at the dyadic level. Another wall dimension is emerging now, thanks, for instance, to my colleague Susan Dickers, of group hyperscanning. So Susan recorded, for instance, classrooms showing how interbrain synchronization can be associated with joint attention, and she will speak, I guess, about that in her talk. Weis Hong in UCLA also show that you can approach interbrain synchronization not only in human but also in animals, and so using cellular recording in mice, they show a even more tangible and demonstration of those interbrain synchronization at the neural level, which is great from a mechanistic point of view. And you'll see over the years, like it has gained a lot of momentum since the first paper of Reed Montague in 2002. Now we have completely skyrocketed the number of publications of hyperscanning and interbrain synchronization. I put voluntarily for the joke the 1965 because they were like a science paper with hyperscanning recording per se, but for telepathy, which was never replicated, and explain maybe 40 years of absence of multi-brain recording afterwards. But anyway, so now it's getting more momentum. So we have written with, like, for instance, Alejandro Perez and also Paulo Barraza, some guidelines to set up your own hyperscanning EEG booth. And we have also, because multi-brain neuroscience needs also software and standard, I've initiated HyPipe, a hyperscanning Python pipeline, which is an open source library to facilitate interbrain synchronization analysis and hyperscanning analysis. So the future is to go beyond the correlation, and so Giacomo Novembre with Gianetti recently put in ticks that this move to causality is linked to multi-brain stimulation. So instead of just measuring to actually perturb the brain, whether by transcranial magnetic stimulation, sensory, motor stimulation, or optogenetics in mice. And Giacomo already demonstrated that in human in 2017 with a motor task and showing how stimulating in phase or anti-phase modulates the behavior of the subject at the inter-individual level. And more recently, Yang and colleague show in Nature Neuroscience how in 3 mice stimulated simultaneously, you can create dyads that are more socially interacting by putting them synchronized at 5 Hz. So you can see here on the right part, while the third mice is at 25 Hz. So like inducing in the triadic context dyad that are more prone to interact than with the third one. So beautiful demonstration that you can have a causal impact on social behavior with interbrain stimulation. But we show also that, well, causality is also at reach with other things than just stimulation. Hyper-scanning in a classical sense can give access to direct causation using feedback loop, for instance. So even Reed Montague in his original paper in 2002 talk about these cross-channel feedback loops that allow people to get the feedback on the interbrain synchronization, and that has already been applied, for instance, by Duan and colleague. So brain-computer interface that integrate 2 brains' activities simultaneously. We can also approach structural causation using human-machine interaction and more classical social neuroscience paradigm. So for instance, if we take the human dynamic clamp that we developed with Scott Kelso and Emmanuel Tognoli, where a human is interacting in real time with a virtual partner, we can track in the brain the neural networks that are associated with self and other, and self and other. And so, for instance, here you can see the motor cortex is associated with self, while the visual cortex is more associated with other. But what is interesting is that the right temporoparietal junction that we saw in the co-ownership of action and as a neural hub between brains in spontaneous imitation actually is the region that integrate both self and other neural networks related to the ongoing behavior. So thus explaining maybe why this appear as a hub in hyperscanning experiments. So kind of mechanistic understanding of the phenomenon despite not implementing stimulation per se. One can argue that it's sensory motor stimulation though, but well. So to finish, we can have even logical causation and explanation through counterfactual demonstration. And by then, I mean just mathematical model or in silico hyperscanning. In that case, you can simulate 2 brains in interaction and virtually connect the motor region of one with the visual region of the other and create this perception action loop. And what we show by doing that is that we have the real connectivity, the real anatomy facilitate interbrain synchronization. So that's explaining that actually, evolutionary speaking, the way the human connectome is organized tend to facilitate the resonance through perception action with another person. Another message that is also interesting, but less about the mechanism, but more about the experimental control, is that in absence of coupling and in absence of this anatomical structure, we still observe a residual synchronization, thus showing that what is interesting in multi-brain neuroscience is not the observation of interbrain synchrony, but how these interbrain synchronizations are modulated by the task and by the context or the group. So you will always observe interbrain synchronization in a way, because there is no zero baseline, except if you're recording 2 random number generator. So just to wrap up, this show that having a shared similar anatomy tend to facilitate this interbrain synchronization, and this open up some question regarding the etiology of autism and how, for instance, the neurodiversity and the variation of anatomy observed in autism can explain the lack of propensity to interact and to synchronize with other people because of this perception action coupling. So that's kind of short, but you have an overview, and here are the take-home message. So I'm looking forward for your questions, and don't hesitate to contact me at guillaume.juma@ptsp.team or on Twitter. I'm pretty active, so don't hesitate to follow. Thanks.
Hi, I want to thank the organizers for including me in this symposium. I do have to admit that I still find it a little weird to be giving a talk to my computer without a live audience. And I guess that just like comes to show how much we rely on face-to-face interactions. That's in stark contrast to the fact that we know very little about how the human brain supports everyday dynamic social interactions, let alone outside of laboratory environments, whether it be in person or virtually. And that's because for decades, we've used a laboratory model of human cognition where we assume that the same perceptual mechanisms apply to the lab and to real-world situations. And for good reason. It has allowed us to overcome critical challenges related to experimental control and data reliability. But it also means that, for example, social communication disorders like autism spectrum disorder have been mostly studied in the absence of dynamic social communication. Whereas we know that the expression of social deficits varies as a function of social dynamics that can be stemming from the social task as well as the social partner. And especially studying the latter might have clinical relevance because it may help researchers understand why caregiver-led interventions are often more successful, for example. So we, like many others, have proposed a model where laboratory studies and real-world studies enter in somewhat of a symbiotic exploration-confirmation dialogue with one another, right? So you might go into the real world and address questions that you can't address inside the lab, but then because your data is messy and less controlled, you go back into the lab to really figure out the details of some of your findings. So to make such a real-world neuroscience approach possible, I work with artists and scientists and educators in a series of hyper-scanning studies where we collect data, EEG data from dyads, but also from groups of people. So for example, in one study led by Diego Reinero, we asked whether interbrain synchrony might predict collective performance, building on questions related to collective intelligence. So researchers and companies like Google have tried to uncover the perfect combination of ingredients for a successful team, but it's still an open question as to what factors really predict team performance. And so we took a neuroscientific angle to this and measured EEG from 44 groups of 4 people who had to complete the same set of problem-solving tasks either as a team, so collectively, or individually. And then we find that interbrain synchrony decreased less over time among team members than for individuals. And more interestingly, we find that interbrain synchrony predicted collective performance for teams, but not for individuals. And that speaks to the social relevance of, possible social relevance of this interbrain synchrony measure. And then we also asked whether interbrain synchrony might predict collective performance better than self-report. And we see that interbrain synchrony continues to predict performance after adjusting for self-report and other behavioral measures. And so this last finding, to me, is probably the most crucial one because it provides ammunition to us people who go out into the world and do these very convoluted and messy experiments, putting headsets on people in classrooms, etc. Because it shows that possibly there's actually value to this. And it's not just that it might provide more information about behavior, it's also that it's an implicit measure, right? So for example, we have studies where we go into the classroom and it's very disruptive to then ask kids to, tell us how they're feeling or what they're learning every step of the way during their regular class. So just to illustrate, we have partnered with classrooms for a full year and recorded brain activity of the kids throughout the school year as they were engaging in their regular class activities. And we found that even though here you can see how they're super, busy with the headsets and the EEG, when they went into their regular classroom activities, they actually seemed to forget that they were wearing these headsets, allowing us to study the brain basis of naturalistic interactions. We asked whether interbrain synchrony during class was related to classroom engagement and classroom social dynamics. And so to jump to the summary, we see that interbrain synchrony is predicted by class appreciation, teacher likability, focus, social closeness, empathy, and group affinity, among other factors. And so now these factors seem pretty diverse. And so it would be very nice if there was some sort of overarching, like some sort of unified mechanism that could explain why all these factors predict interbrain synchrony. And so one overarching, factor that all of these have in common is that they might predict shared attention, which in turn might predict interbrain synchrony. But shared attention is, a broad construct, to say the least. So it may be useful to impact social situations just a little bit further to really try to get at how neurocognitive processes might support the dynamics of real-world communication, right? So each person comes to a social interaction with their own personality traits, neural architecture, mental states, and priors about the situation, and the similarity with others in these factors may already predict interbrain synchrony, even in the absence of social communication. And we know that lab studies that the same is true for external stimuli, right? So ranging from tones to movies. But then the most relevant to our purposes, of course, is how social behavior, which may help establish a common ground, can also affect synchrony and social closeness. And so now we can study how each of these components might independently predict synchrony. For example, for our school data, the teacher is an exogenous stimulus with a temporal structure. And if kids are not engaged, there's also no neural entrainment with the teacher's speech, and therefore there is no interbrain synchrony. In the teacher's dream world, of course, all the kids are super engaged and their brainwaves are perfectly locked to their speech, causing perfect interbrain synchrony. But in the real world, there's obviously individual variation where some kids are more engaged than others. And these are also predicted by by social factors like empathy, etc. And so these different kinds of social situations then allow us to test different boxes in the schematic I drew for you, right? So we've talked about teams and school situations. We are also studying psychotherapy, families in different socioeconomic situations, and crucially, autism spectrum disorder. For example, together with the Sackler Institute and with Northwestern University, we have projects exploring how social situations can affect synchrony in ASD. So we collect EEG and video during adult-child interactions, where we compare kids with ASD and kids that are typically developing as they're interacting with a caregiver or with a clinician, either playing with a toy or while having a conversation. And so we see here that both eye contact and synchrony increases when there is no toy present, right? So that's object engagement is when there is a toy. Crucially, this increase is stronger when kids are engaging with a caregiver. And in the brain, it's also stronger for typically developing kids. So it seems to matter, unsurprisingly, that when you study social disorders, to study it in the social context, it matters who you talk to, it matters what you do, and it also matters who you are. And so the latter speaks to the non-social contributor, so to speak, to interbrain synchrony in social interaction. So there's a growing number of researchers who are understanding that there's large variation with respect to neural architecture and how social deficits might express themselves. And the one possible cause could be differences in oscillatory patterns, causing different patterns of engagement and attention, which may cause differences in synchrony indirectly. In another line of related work, we use hyper-scanning neurofeedback. So about a decade ago now, my partner in crime and interactive media artist, Matthias Oostrich, and I developed a software that allowed us to record data from multiple people and translate interbrain synchrony into different outputs, like, for example, the extent to which two heads merge in and out of each other. So this is a neurofeedback game where dyads can try to sync up their brainwaves as much as possible. And then after 3 minutes, they can compare their synchrony score to people that went before them, or they can try out different synchrony strategies. This game has proven super, fruitful and valuable for outreach in science museums and festivals. And it has also enabled us to test the viability of collecting data under more naturalistic circumstances and to crowdsource neuroscience data, where we use interactive installations in museums like the Mutual Wave Machine, for example, to collect data from lots of people. So here in the Mutual Wave Machine specifically, people are immersed in an environment where their synchrony is translated into light patterns. We have had over 5,000 participants. It's currently on exhibit at the National Museum of Modern Art in Lisbon. And I'm going to show you some data from the Benaki Museum in 2016, where half of the people were told that more light was translated into, or related to more synchrony, and half of the people were actually not given this information. And this was based on the hypothesis that people might be motivated by external, triggers like neurofeedback to engage more and thus synchronize more. And we tested this in addition to social closeness, personality traits, and mental states. So now we're dealing with less than suboptimal recording conditions, one might say, euphemistically. And so this is kind of offset by the fact that we have tons and tons of data sets. So we can also delete data sets that were not up to the data quality standards. And we also used interbrain synchrony metrics for which we know that they are likely less sensitive to common noise in the environment. And so we find that dyads who reported higher social closeness showed higher overall synchrony, just like we see with teacher-student closeness or student-student closeness in the classroom. We also see that dyads with lower personal distress show higher overall synchrony. So this is the same measure and the same findings in the classroom with empathy. And interestingly, we also find that only the neurofeedback group shows an increase in synchrony over time when you compare the second half of the interaction to the first half of the interaction. And so this finding relates back to what we saw in the team study, right, where we found that synchrony decreased over time, but a little less so for those who were socially engaging than those who were not. And so this is possibly explained by a process where, people enter this environment with a lot of, exogenous stimulation, which causes, synchrony just by virtue of the fact that they're entrained with the same stimulus. But then a group who thinks that there's, or believes that there is a relationship between what they're seeing and how much they're engaging, actually pushes them to also, on top of that, engage in social interaction and joint action, which may help increase synchrony above and beyond what a shared environment or a shared stimulus does. And so if that is true, it might be promising for using hyper-scanning neurofeedback interventions to help improve social communication, and especially with people who might be more inclined to respond to computer-mediated training of social interaction, which is known for kids with autism. To make this possible, we've developed software, and I am in front of my own hashtag, hybrid harmony. And the hybrid harmony allows you to record EEG from multiple people. It uses LSL, last streaming layer, to port the data, it pre-processes the data for you, et cetera, and it allows you to visualize interbrain synchrony. But most crucially, it also allows you to use different connectivity metrics to analyze your data, because people use very different kinds of ways to compute synchrony. I've really kind of glossed over that in my talk, but there are almost as many metrics as there are studies, and different metrics might also differentially relate to different social factors. People are using these different metrics, and now I need to move away from in front of my friend Guillaume, who has spearheaded the effort of the hyperscanning Python pipeline, HyPipe, with the aim of unifying the fields in the way that we look at and analyze data, and that when we make our choices for our different synchrony metrics, that those choices are actually well-founded, and not just because some might, generate the results that you want and some might not. Anyway, I'm at my time, and I just wanted to show you a subset of all the people that are involved in our projects. As you can imagine, it's many. All right. Thank you so much for listening.